Hello everyone, welcome back to my channel. I'm Poulani and today we are back with another video on immunology, which is on MHC or Major Histocompatibility Complex. We will understand what is MHC Class 1, what is MHC Class 2, what are the differences between them in this video. So let's begin. So guys, you all must be aware of organ transplants and all. Okay, now see what happens whenever there is organ transplant. Okay, we transplant a particular organ. Say suppose you say liver transplant. Okay, so when the liver is taken from the donor and when it is transplanted into the patient's body, then what do we look into? Okay, apart from the matching, what do we match? Okay, exactly what do we match? We match the histocompatibility. Okay, whether the tissues are compatible enough to match. Okay, we match the MHC receptors out there. So what happens in our body? Okay, generally like MHC1 and MHC class 2. Okay, this class 1 and class 2, these two are present. Now what happens, the MHC class 1, they are present in almost all the cells, and MHC class 2 is only present on the antigen presenting cells or your APC cells, which includes your macrophages, dendritic cells and your B cells. Now suppose you think that one particular cell of our body is damaged or disease, okay, and we are unable to repair it at that moment, and if it stays in our body, then it will only cause harm. So to lyse the self cell, okay, to lyse or to break down or to put it into death, okay, we use MHC class 1. Okay, so MHC class 1, what do? They provide signal. Okay, they go with the help of your TCR and all. Okay, it goes and it lyses the cell. So that is the work of your MHC class 1, and I will explain that too in detail with you. And the work of MHC class 2 is actually to entrap the antigen, okay, or the foreign substances which is coming inside the cell, to entrap it, okay, to engulf it, to phagocyte it, okay, and to break down, make it into fragments, okay, and then to place it and present it, okay, and present it to your T cells. So that is the work of your MHC2. In very short, I have told you. So now what happened in history wise, if we see, George Snell, okay, he discovered the first component of the MHC through their role in rejecting the transplant in mice, okay, and he created the word histocompatibility. Now see next, around a decade later, okay, for around years later, what happened? Another scientist, he uncovered the first compatibility antigen in humans, okay, so he uncovered the first compatibility antigen, this process in humans. Then again the other scientists, okay, in the 1970s, okay, what happened? He provided the first indication that the immune reactions are controlled by the MHC gene or immune response gene. So this thing was made very clear to us. So what happening? The rejection, okay, that the body is facing, okay, or the like the guinea pigs and all that, whatever, like mice, okay, all this onco mice and all, whatever is taken for the experiment, okay, they are the rejection that the cells they are facing, okay, it is due to nothing but the MHC genes that is present. So they play a very major and important role in the body's immune system. So that's why we are reading it. Okay, now see next what is written. Basic terminology is histocompatible. So histocompatible means that the transplanted tissue is successfully accepted as self. Okay, self, that means it is compatible. Another is the histocompatibility antigens. Okay, it means that the rejection of foreign tissue is the result of an immune response to cell surface molecules. Okay. Next you see histocompatibility complex, okay, a region of multiple loci, okay, loci of a gene that play a major role in determining whether the transplanted tissue is histocompatible or histoincompatible. Okay, now see next, major versus minor. So major histocompatibility complex MHC causes your rapid graft rejection, and minor histocompatibility complex causes your slow graft rejection. Now HLA, now this this MHC, okay, it is named differently in different organism. Now see HLA, so human leukocyte antigen or HLA, it is MHC antigen in human beings, and next is your H2, which is again important, this MHC antigen in your mice. So I hope this slide is also clear to you. Although the arrangement of gene is somewhat different, guys, MHC are, they are arranged, okay, into regions which is encoding classes of molecule, that is your MHC1, MHC class 2, okay, and MHC class 3. Abhi dekho. The major histocompatibility complex is a group of genes coding for a set of host cell surface molecules that bind to the peptide fragment derived from pathogen, and they display them on the host cell for the recognition by the T cell. What is happening whenever a foreign pathogen, say suppose, okay, and virus is entering in our body? Okay, now what happens? That is an antigen. So to give the signal, what is happening, the APCs, okay, they're like macrophages and all, okay, they're going and entrapping it, okay, they're engulfing it. Now they're breaking down it into pieces, okay. Then, okay, inside there is your ER, okay, out there what is happening? There will be two things, okay. First is your receptor, okay, the receptor will be there, and on that receptor that antigen fragment will be there. So before, when there will be in MHC2, if I say before, antigen, okay, there will be a clip kind of thing that will be covering the area, but once the antigen is recognized, okay, they will put it on the receptor and they will place it like that on the surface of the macrophages or your APC molecule, any of the APC cells. Now what is happening when they will see, okay, when the T cell will come and see, they will recognize it, and gradually they will, they will release the signals, and then, the B cells, okay, they will get activated, or whatever they feel like, whatever will be the process, okay, later process will be continued in this manner. Now see, these are present in almost all human cells, but first discovered on the surface of your leukocyte or WBCS. Hence in human, the MHC coded proteins are also known as the human leukocyte antigen cells, okay, antigen or HLAs. So it is found in all cells, that what I told you just now, but since it was first discovered in the leukocyte of human beings, so we name them as human leukocyte antigens. Now see next. MHC molecules serve as a unique identification marker for every individual, as the genetic sequence of MHC gene is different for every individual, okay, it is different. That's what I told you already, that it is different, it is somewhat different for all the individual. Okay, now the transplantation of graft, okay, graft means a particular piece of organ that we need, okay, the recipient mounts and the immune response against the graft MHC molecule and vice versa, okay. Now see, greater is the immune response, greater is the rejection of graft, or you can say graft rejection. Now the acceptance or rejection of the graft is directly dependent on the MHC molecule of the graft and the recipient. That's what I told you, that it should match with the MHC molecule. Both the MHC molecule must match, only then it will be accepted, or else it will get rejected. Okay, now see, in humans the HLA complex coding for MHC molecule are located in the short arm of chromosome number 6. So that is their location. Okay, so I hope this this introduction, this slide 2 was also about introduction, so it is also clear to you. So if you are liking my video, then don't forget to like to this video, and don't forget to subscribe to my channel. Now look at the diagram of MHC1, guys. Out here you see alpha 1 and alpha 2. Okay, in between them the peptide binding cleft is there. So out here the antigen will come and bind. Now see, they are the membrane distal domains. Next you see alpha 3, which is your membrane proximal domains, okay, that is your, and again you can see the beta 2 microglobulin, okay, it is somewhat supporting the structure. Now see, there is transmembrane segments there, and then there is your cytoplasmic tail, okay, and you can see the sulfide bonds also in between each of this. Now we will understand, okay. Now see, MHC class 1, okay, it is about 2000 kbb in length and comprises of your 3 class gene, which is known as HLA-A, B C. Genes which code for HLA-A, HLA-B and HLA-C proteins. Okay, these genes they code for ABC proteins. Now see, each one is capable of forming your alpha chain of MHC class 1 molecule, and the alpha chain they are polymorphic, okay, and transmembrane. What do you understand by the term polymorphic? Polymorphic means nothing like a same particular compound, okay, they are changing, they are showing different morphology, okay, different shapes देखने को हमें मिलता है. अभी देखो, alpha 1 and alpha 2 domains they form the peptide binding cleft. ठीक है, जो ऊपर है peptide binding cleft form हो रहा है. नीचे देखो alpha 3, okay, alpha 3 interacts with your CD8 receptor एंड ऑन द टी सेल. ठीक है, ये जाके क्या करता है, CD8 रिसेप्टर जो है टी सेल में उसके साथ जाके, हमेशा याद रखना जो MHC फर्स्ट है क्लास 1 है वो CD8 से जाके इंटरेक्ट करेगा, और MHC जो 2 है वो CD4 के साथ जाके वो इंटरेक्ट करेगा. ठीक है, देखो now beta chain doesn't shows any more polymorphism, there is no more polymorphism. ठीक है, वो अपने एक ही शेप में रहता है. ठीक है, then there is one folded domain out there and they stabilize the class 1 molecule. Stabilization show कर रहे हैं. ठीक है, देखो next, okay, and it is seen on the gene chromosome number 15. Okay, this is also important point. Now see, the MHC class 1 proteins located on the surface of all nucleated cells, okay, except your sperm cells, okay, like your eukaryotic cells हैं. ठीक है, they are true nucleated cells. तो MHC class 1 जो है ये हर एक like in human cells you see, in eukaryotic cells of you say of other organisms, ये हर एक सेल में जो है प्रेजेंट होते हैं. न्यूक्लिएटेड जो ट्रू न्यूक्लिएटेड सेल है उसमें प्रेजेंट होते हैं अपार्ट फ्रॉम द स्पर्म सेल्स. नाउ सी, they are absent in RBCs also, they are not present in RBCs, though they are particularly found in your leukocyte, that is your WBCS. And next they are present in the peptide antigen. ओके, दे प्रेजेंट द पेप्टाइड एंटीजन. जो पेप्टाइड एंटीजन होता है उसको प्रेजेंट करते हैं किसके सामने, CD8 T सेल्स के सामने. ओके, सो दैट इज ऑल अबाउट द स्ट्रक्चर ऑफ MHC class 1. I hope this slide is clear to you. Now let's look at the structure of MHC2 molecule. Okay, you can see alpha 1, beta 1, alpha 2, beta 2. Okay, both are anchored into the cell membrane. Okay, so it is not hanging, like it is not like the structure of MHC1. So it is you can see, okay, they both are different in structure wise also. And what is happening, okay, see in these regions, okay, there is a peptide binding cleft out there. Okay, and again it is the the cytoplasmic tail is again out there, okay, which is similar. Now see, it is about your 1000 kbp in length, and it comprises of 3 region. Again, like your MHC1, it has 3 region. Okay, it has DP, but in this case they have DP. Okay, in MHC1 there was your HLA, but in this case it is DP. So it is DP, DQ and DR genes which is encoding for your DP, DQ and DR proteins. Okay, now see, each of them are capable of forming the alpha chain and the beta chain class 2 molecules. Okay, now see, beta and alpha chain, okay, both are anchored into the cell membrane. That's what I told you just now. They both are polymorphic, means they can change their shape. Okay, they can change their morphology. Next you see the MHC2 region, they also contain certain other non-classical genes like your DO, DM, LPM, okay, and your TAP, which is your transporter associated antigen processing, that helps in your antigen processing and presentation. So which is your unique thing. देखो अभी जो है MHC2 protein, okay, they are located on the surface of the antigen presenting cell. So antigen presenting cell, you already know the APC cells, okay, which are your macrophages, which could be your dendritic cells or your B cells. Okay, they will go and present the particular thing and in front of your T cell, and further the T cell they will release their cytokines and all, and then they will give their signals to the B cell and all. Okay, then the entire thing will be happening. Okay, now they present what? They present the peptide antigen to the CD4 T cell. Okay, so I hope this slide is clear to you. Look at the differences, okay, like MHC class 1 and MHC class 2. What are the difference? If you just understand these differences and you keep it, okay, in your mind, then your work is half done. Okay, now see, MHC class 1 and MHC class 2, where are they present? MHC class 1, they are present on all the nucleated cells, okay, but except your RBCs, okay, sperm cells, okay. And next you see MHC class 2, that's what you already know, APC, okay, antigen presenting cells. Next is your peptide antigen, okay, peptide antigen, it will present to the CD8 T cell, and in this case it will present to the CD4 T cell, okay. Next you see nature of peptide antigen, okay, it is your endogenous or your intracellular, okay, which could be a viral or tumor antigen, okay, that's what you know, okay. Like I told you, MHC class 1, okay, they are acting on the self cell, like, you know, like their own cell which has got damage, okay, like the tumor antigen, you know, the proto-oncogenes, they are present in our body, right? But it may, you know, sometime it may cause your trigger, you know, it may trigger the tumor in our body. So in that case what happens, हमारी जो बॉडी की सेल है, ठीक है, हमारी जो बॉडी की MHC क्लास 1 के जो सेल है वो जाके क्या करेंगे, उस पर्टिकुलर सेल को डिस्ट्रॉय करने के लिए जो MHC क्लास 1 को है इसे एक्टिवेट कर देंगे, सो दैट यू नो फर्दर डैमेज ना हो, ठीक है, हमारी बॉडी में ना हो. दूसरा देखो, एक्सोजेनस है MHC क्लास 1, डेफिनेटली बाहर से आ रही है, फॉरेन पार्टिकल पैथोजन. नेक्स्ट देखो, पेप्टाइड एंटीजन का साइज देखो, 8 to 10 अमीनो एसिड लॉन्ग, दूसरा आपका 13 to 18 अमीनो एसिड लॉन्ग, ठीक है. दूसरा देखो, एंटीजन प्रेजेंटेशन पाथवे, इसका है साइटोसोलिक पाथवे, ठीक है, दूसरा है आपका एंडोसाइटिक पाथवे, ठीक है. पाथवेज के बारे में भी मैंने जो है ऑलरेडी वीडियो बनाया है, अगर आपने नहीं देखा तो प्लीज़ गो एंड वॉच दोस वीडियो ऑल्सो. एंड आई हैव टोल्ड यू नो कि अगर आपने प्रीवियस के कोई भी लेक्चर्स को मिस किया है तो आपको आगे के वीडियोस में समझने में मुश्किल होगी, पर मैं आशा करती हूँ अभी तक मैंने जो सब चीज़ें आपको समझाया है, राइट फ्रॉम स्ट्रक्चर, ओके, एंड देयर यू नो की डिफरेंसेस, ओके, इट इज़ क्लियर टू यू. नाउ सी नेक्स्ट, ओके, नेक्स्ट इज योर पेप्टाइड बाइंडिंग साइट. इसमें क्या हो रहा है, अल्फा 1 और अल्फा 2 ग्रोव में हो रहा है, एंड MHC क्लास 2 में क्या हो रहा है, अल्फा एंड बीटा 1 ग्रोव में हो रहा है, ठीक है. नेक्स्ट CD4 और CD8 बाइंडिंग. सो अल्फा थर्ड जो है, ठीक है, ये जाके बाइंड कर रहा है किससे, CD8 मॉलिक्यूल से, जो T साइटोटॉक्सिक सेल्स में हो रहा है, साइटोटॉक्सिक T सेल, ओके, दैट इज टू बी मोर स्पेसिफिक. दूसरा आपका जो CD4 और CD8 बाइंडिंग, मतलब ये बेसिकली CD4 बाइंडिंग जो है ये बीटा सेकेंड जो है वो जाके कर रहा है कहाँ पे, टू CD4 ऑन हेल्पर T सेल. Okay, I hope this differences is also crystal clear to you. So guys, that's all for today's short and crisp video. I hope you have enjoyed this video. If you find any, you know, if you have any doubts or you know you have any queries regarding anything whatever I have taught in today's class, then please do let me know in the comment section. And you can also suggest me the videos that you want from me and you want me to make after I complete the immunology part. Okay, please let me know in the comment section. And if you like this video, then please do hit the like button to this video and share it with your friends. Okay, spread it to more and more people. Okay, bring more and more subscriber to my channel, so I will get motivated enough to present you better and better lectures. Okay, so guys, that's all for today's class, and keep making notes, keep studying hard. Okay, all the very best. If you are having exams in front, then all the very best from my side for that examination point of view also. So guys, that's all for today's class. Bye bye, take care. Okay, good night.